from Channel for All. This is Get Curious, exploring everything under the sky and above it.
All right, today we are diving deep into the code of life itself. We're going to explore biotechnology and genetics, and we're going to do it through a really fascinating lens, the quest to build the perfect champion.
So picture this, a thoroughbred racehorse. You know the type. It has that explosive speed, those powerful muscles, and just an unbreakable will to win. It's the perfect athlete. But how does a creature like that come to be? Was it just born that way, or was it made? And the bigger question is, could we do it again, but this time completely on purpose? That's what we're going to unpack.
Now, to even think about answering a question that big, we first have to understand the blueprint, right? We've got to go right down to the cellular level and learn how to read the instruction manual that builds every single living thing on this planet. And that instruction manual, well, it's called DNA. The easiest way to think about it is like a master cookbook for our champion horse. Every single tiny instruction, from the color of its coat to the sheer power of its heart, it's all written right there in this incredible molecule. Okay, so if DNA is the whole cookbook, then genes are the individual recipes inside it. You've got a recipe for speed, a recipe for muscle mass, another for endurance. You get the idea. Each gene is just a specific chunk of DNA that codes for one particular trait. These are the absolute basics of heredity. But here's where it gets interesting. Recipes can have variations. In genetics, we call these variations alleles. A dominant allele is like a bold all-caps instruction in the recipe. It always gets followed, even if it's only there once. A recessive allele, on the other hand, it's more like a quiet suggestion. It only gets used if you have two copies of it with no dominant one there to talk over it. Which makes you wonder, right? If an animal can carry a hidden recessive trait without actually showing it, how can we possibly predict if their kids will get it? You know? How do you even begin to calculate those odds? Well, lucky for us, we have this wonderfully simple tool called a Punnett square. It's basically a genetic probability calculator. So let's say we cross two parents who are both carriers. That means they each have one dominant allele, we'll call it big A, and one of those hidden recessive ones, little a. You just combine the letters from the top and the side, and you can see all the possible outcomes for their baby. And when you run the numbers, the results are pretty clear. There's a 25% or a 1 in 4 chance that the offspring gets two dominant alleles. There's a 50% chance it ends up being a carrier, just like its parents. And then there's a 25% chance it gets two recessive alleles, which means finally that hidden trait is going to show up. Okay, so we can read the code. We can even predict the outcomes. But what if we want to do more than just predict? What if we want to actually get in there and rewrite the rules? This, my friends, is where biotechnology enters the picture. The classic method, the one we've been using for thousands of years, is selective breeding. It's the oldest trick in the book, really. A horse breeder sees two super fast horses and thinks, hey, I wonder what would happen if I bred them together. Then they take the fastest of the offspring and do it again and again and again, hoping to make that speed trait stronger over many generations. It works, but man, is it slow. And there is a pretty big trade-off. When you focus so hard on just one set of genes, you start losing all the other ones. The overall genetic diversity of the population shrinks, and that can make the whole group way more vulnerable to a single disease wiping them out down the line.
The modern approach, though, is way more precise. It's called genetic engineering, and it's not about just hoping you get the best traits. It's about choosing one specific gene and inserting it directly into another organism's DNA. It's like a surgical strike on the genetic code.
And trust me, this is not science fiction. This happens every single day. Take insulin for people with diabetes. Scientists take the human gene for making insulin, they pop it into the DNA of some harmless bacteria, and then they just let those bacteria multiply like crazy. The bacteria become these tiny living factories just pumping out huge amounts of life-saving human insulin. It's an absolutely incredible use of this tech. So if we can read the code, and we can even rewrite the code, what about making a perfect copy? Let's go back to our champion racehorse. We've got a winner. How do we guarantee we get another one just like it? The answer might just be cloning. Cloning is the process of creating a genetically identical copy of an organism. Not just kind of similar, we're talking a perfect genetic duplicate. It seems like the ultimate way to preserve those winning traits, right?
Well, it's not that simple. Cloning presents a huge dilemma. On the one hand, yeah, you could preserve the genes of a champion, or create animals for medical research, or maybe even help save an endangered species. But the flip side is pretty serious. The success rate is often incredibly low. Plus, it reduces genetic diversity even more, creating a population of identicals that could all be wiped out by the same disease. And, of course, it opens up a massive can of worms when it comes to ethics.
And that brings us to the final and maybe the most important piece of the puzzle, because it turns out even a perfect genetic copy might not be the magic bullet we're looking for.
So let's just ask the big question straight up. If we take our champion racehorse and we create a perfect genetic clone, is that new horse guaranteed to be a champion too? The answer is nope, not exactly. The clone has the exact same genetic blueprint. Sure, the same set of recipes. But that blueprint is only half the story. The environment, literally everything that happens after that clone is created, plays a massive role in how things turn out.
Here's why a clone isn't a carbon copy. Yes, the genes are identical, but even in the womb, tiny random little things happen during development that make a difference. And most importantly, after it's born, the environment will be totally different. The training program, the food it eats, its relationship with its jockey, none of that can ever be perfectly replicated. So you can clone the genetic potential, but you can't clone the life that actually unlocked it.
And this is what brings it all back home, to us. Because understanding our own genetic code, our own personal instruction manual, is every bit as complex. Our genes don't decide our destiny either.
And that's exactly why fields like genetic counseling are so critical. It's a service that helps people read their own genetic code to understand if they're at risk for certain inherited diseases, and then helps them figure out what to do with that powerful and, let's be honest, sometimes scary information.
Which leads us staring at one final massive question. We have the tools now. We can read, we can rewrite, and we can even copy the fundamental code of life. We've basically gone from being subjects of evolution to its authors. So, what should the rules be? Who gets to write them?